Welcome, and thank you for attending IDEX Health and Science's e-Symposium on automating analytical workflows for bioproduction. Increasingly, biopharmaceuticals in their many forms are positively affecting patient outcomes. However, analytical testing throughout the biologic drug development and production process continues to be challenging. IDEX Health and Science, being a global leader in fluidic technology innovation, is ideally positioned to provide analytical application and engineering solutions for bioproduction workflows. Bridging the technology gap between cell culture processes and analytical instrumentation, IDEX partners with equipment manufacturers to develop solutions for challenging workflows, provides core technologies along entire flow paths from sampling to detection, and supplies chromatography and bioprocess PAT application expertise. Streamlining the analysis of biopharmaceuticals by automating workflows significantly improves insight into production processes, which, in turn, can accelerate product development timelines. IDEX's product development model involves ongoing communication with biopharmaceutical scientists and partnering with instrument vendors to provide novel solutions for even the most challenging analytical workflows within this industry. As a market leader in fluidics, with application expertise that spans automated bioreactor sampling, sample preparation, chromatography, and mass spectrometry, IDEX is the perfect partner for instrument vendors committed to supporting bioprocess PAT. IDEX provides holistic solutions using a multitude of fluidic technologies as building blocks. We problem solve through a combination of engineering partnership, application expertise, and workflow integration, with our customers ultimately benefiting from easy-to-use and differentiated products that enable faster time to market. Biomolecule production in any form is complex, with numerous process inputs, control parameters, KPIs, and CQAs being monitored using a variety of sensors, analytical instruments, and laboratory assays. Currently, many key bioprocess analyses are performed offline and retroactively, resulting in extended turnaround times for actionable process data. Integrating and automating these processes can shorten development times and lower costs. The sampling to analysis workflow shown here highlights where some of IDEX's many fluidic solutions can be used to support bioprocessing PAT. Bridging the bioreactor to analyzer technology gap has been an underlying theme of our work at IDEX, where we are striving towards integrating biopharmaceutical processes with analytical technologies such as mass spectrometry to provide insight into critical quality attributes of complex biomolecules during production. Bringing MS and other analytical tools closer to the cell culture process to provide real-time analysis remains an important area of focus. Being a global leader in rotary shear valve technology, IDEX uses these movable manifolds as the centerpiece of many integrated fluidic solutions and subassemblies to provide unlimited options for customization. In this example, a movable manifold forms the core of a multiplexed analytical workflow that draws up sample from a tube tray, passes this through a detector flow cell for analysis while the fluid lines are washed and cleared with filtered air. The flow cell is then rinsed, while sample from a second tube tray is drawn up and passed through the flow cell. IDEX's rapid prototyping program produces custom subassemblies similar to the one shown here, within timelines that can be as short as 4 weeks. The utility and flexibility of our valves will be demonstrated by a number of workflows examples to follow. IDEX also remains the leader in solvent degassing, with many vendors' HPLC systems relying on our degassers. Our constant performance vacuum control has improved solvent degassing performance by reducing pervaporation and optimizing efficiency at any flow rate. Also, the evolution from conventional lumen-based coil tubing to a flat film membrane form factor has further expanded the use of IDEX degassing technology. In addition to being widely deployed for LC, the unique properties of flat film membranes have enabled the development of innovative applications that extend beyond chromatography, as these membranes are readily adaptable for a wide variety of low volume applications, sterilizable by steam or gamma irradiation, and operational at pressures between -14 and 30 psi. My colleague, Chuan, will describe one such application recently developed at IDEX that addresses several challenges associated with automating sampling in bioprocessing.
Before we dive into the specifics of this application, it is important to first highlight the similarities and the differences between the porous membranes currently being used for filter and our flat film membrane technology. 0.2 micron porous filters are the bioprocessing industry standard for gas and liquid media sterilization and bioreactor contamination control. These filters have a pore size designation, such as 0.2 micron, which means that the filter will retain particles with a diameter of 0.2 micron or greater. These porous filters have a high gas flow rate under pressure differential, but particles or foreign objects less than 0.2 micron in diameter can still pass through. Porous media can also be blocked by solid, which reduce filtration efficiency. IDEX flat film membrane technology uses a non-porous material. This film only allows gas phase molecules to permeate, and no liquid or solid can pass through. This membrane provides an excellent sterile barrier and has a consistent gas permeation rate sufficient for moving and degassing liquids. The following examples highlight various applications where IDEX core technologies have been successfully integrated and are used for automated aseptic sampling, dilution, filtration, purification, desalting, and antibody sample preparation. Aseptic sampling from a bioreactor is critical for preventing ingress of contaminant, thereby maintaining system sterility. Using the unique properties of its flat film degassing technology, IDEX has developed an innovative automated system for aseptic sampling from bioreactors that is simple, compact, flexible, and can deliver samples direct to any analytical device. IDEX automated aseptic sampling solution provides a degassed sample drawn from a bioreactor that can be fed directly to a sample prep device, LCMS, or any other analyzer. This small footprint module consumes less than 100 microliter of sample and can completely automate airline PAT workflows. Short sampling times, even with long sampling tube length, are possible due to parallel priming capabilities. Sample probe fouling can be mitigated by backflushing with sterile air permeating under pressure through the flat film membrane. IDEX has developed a fully automated 4-channel technology demonstration sampling module. This capability is designed for custom integration into cell culture, sample prep, or analytical product platforms. Preventing bioreactor contamination remains a significant challenge within bioprocessing. We have rigorously tested the ability of the IDEX sampling system to prevent contamination during regular sampling intervals. Using a steam sterilized 3-liter fermentation vessel containing nutrient broth to simulate a contamination-free bioreactor environment. Our membrane-based sampling system was able to maintain contamination-free conditions for over 100 days while successfully completing over 200 automated sampling events. Sample carryover was also tested using a highly concentrated caffeine solution in one channel and a water blank in the other channel. No residue sample response was observed after multiple sequential sampling between these two channels. This automated aseptic sampling technology will be on display at the 16th Bioprocessing Summit being held in Boston on August 19th to 22nd. Following on from automated bioreactor sampling, the IDEX inline dilution and the filtration concept shown here integrates two fluidic technologies, namely dilution and filtration, into a single sub-assembly. This automated inline dilution module includes two IDEX compact bioinert pumps to control the dilution ratio, along with a novel sample injector that combines the two fluid streams. With the sample loop filled, when the valve switches, the sample is mixed with the diluent stream and dynamically diluted inline. The diluted sample stream enters the cross-flow ultrafiltration module, which filters out large molecules while small molecules pass through into the collection stream. The IDEX module provides a pipette-free alternative to traditional precipitation and the dilution workflows for automatically cleaning up samples containing large biomolecules. This figure shows inline dilution module performance. Using caffeine standard solution for monitoring, using a UV detection at 280 nanometer, we observe excellent linearity across a wide range of dilutions from 1 in 10 to 1 in 500. This graphic shows the performance of the integrated inline dilution and ultrafiltration modules. It shows size exclusion chromatography result of samples prepared using different methods. The 5 linear point shows protein standard molecule elution times. The blue chase represent a spent media sample diluted without filtration and has peaks at elution times corresponding to molecules larger than 10 kilodalton. The orange trace shows a sample prepared using conventional organic solvent precipitation, followed by centrifugation and the dilution. This method removes large molecules, but also reduces the concentration of a small molecule. Our automated inline dilution ultrafiltration module, shown by the purple trace, removes large molecules, but does not alter the small molecule concentration. This study demonstrates that our combined dilution and filtration capabilities can successfully dilute and prepare samples from complex matrices for small molecule analysis in an automated pipette-free system. Now I will hand over to Craig, who will describe several more fluidic capabilities that IDEX has developed.
Thank you, Chuan. I would like to start off by illustrating how inline protein A sample purification can be integrated into a fluidic workflow. Protein A purification can be utilized to prepare MAB samples for intact mass analysis or for further processing, such as enzymatic digestion or peptide mapping. The addition of inline UV detection allows automated calibration and titer measurements to be performed. By loading variable sample volumes, calibration curves can be created without the need to prepare serial dilutions of your calibration standard. This approach saves valuable time and helps eliminate dilution errors compared to manual sample preparation. This functionality can easily be integrated into other workflows for MAB sample preparation and analysis using the extensive portfolio of IDEX fluidic solutions. Here we have an example of a linearity curve created using variable injection volumes of a human IgG standard with inline UV detection. The resulting curve is highly linear and covers a wide detection range from 2 micrograms to 100 micrograms on column. While this curve was generated using a fixed calibrant concentration, the use of variable injection volumes also allows a wide range of sample concentrations to be quantified. For example, the 500 microliter injection volume could be used to measure a sample concentration down to 4 micrograms per mil, or 4 mg per liter. Correspondingly, the 10 microliter injection volume could be used to measure sample concentrations up to 10 mg per mil, or 10 grams per liter. Desalting, or buffer exchange, is a crucial processing step in bioprocessing workflows to prepare therapeutic proteins and monoclonal antibodies for subsequent chromatographic analysis or sample processing steps, such as enzymatic digestion. We can see that inline desalting or buffer exchange uses the same basic fluidic configurations as Pro-A purification. This functionality can also be integrated into an automated workflow to accelerate sample preparation by eliminating tedious manual centrifugation steps. Another benefit of the inline desalting workflow is the ability to add inline UV detection for real-time process monitoring. This facilitates the optimization of desalting conditions and can provide an estimate of product recovery following desalting. This slide demonstrates the performance of the inline desalting workflow. For this illustration, caffeine was used as a salt surrogate to monitor the desalting performance at 280 nanometers. This allowed optimization to be performed using a fixed wavelength inline UV detector. The inset image in the upper left shows the sample output of the inline UV detector, where the artifact between the two peaks is caused by refilling of the dispense pump after the sample peak. In practice, the sample peak is typically retained for analysis or further processing, while the salt peak is sent to waste. The main image shows an analytical SEC comparison of the sample before and after the desalting step. In this example, sample recovery was approximately 80%, while the salt content was reduced by approximately 80%. While the previous examples have been primarily one-step sample preparation workflows, this illustration shows a multi-step sample preparation schematic for N-glycan analysis. In this case, a commercially available magnetic bead-based kit for N-glycan purification and labeling was adapted to an inline fluidic workflow utilizing multiple capabilities, including sampling, heating, mixing, reacting, separating, and rinsing. These fluidic building blocks were used to successfully denature, digest, label, and elute the labeled glycans from a monoclonal antibody standard for N-glycan analysis. This is another example of utilizing a rotary shear valve as a movable manifold to manage complex workflows. In this close-up of the valve port assignments on what we term our 24/25 valve, short for 24 positions, 25 ports, we can see how the various fluidic functions and reagents can be arranged in a compact footprint to simplify system designs. One of the fluidic capabilities used in the previous example is inline mixing. This functionality is essential for obtaining a uniform mixture when two or more liquids are combined in a fluidic stream. Perhaps the simplest application of inline mixing is sample dilution, as illustrated in this fluidic schematic. In this example, An air segment and half of the diluent volume was aspirated into the holding coil, followed by the sample volume, then the second half of the diluent volume, and a final air segment. This air and fluid segment was then sent to a mixing coil where it was mixed using forward and backwards pump movements. After mixing, an inline UV detector was used to assess the homogeneity of the fluid segment. This is especially important if a portion of the fluid segment is being loaded onto a sample loop. If the fluid segment is not homogeneous, the calculated concentration will be incorrect. Looking to the UV trace to the left, we see a common goalpost shape for each of the sample traces. The two posts in each of the traces are caused by the leading and trailing air segments, while the center bar is an indication of the sample homogeneity. We can see that the center portion of the no mixing trace, shown in blue, has a significant absorbance slope, indicating a lack of sample homogeneity. This sample was sent from the holding coil directly to the UV detector, bypassing the mixing coil. The next trace, in orange, shows the effect of adding 2 cycles in the mixing coil. The center portion of this trace is flat, indicating a uniform sample concentration across the segment. A sample blank, consisting of water and the air segments, indicates that carryover is not a concern at these sample concentrations. With our novel aseptic bioreactor sampling technology, IDEX Health Science can engineer OEM sampling solutions for cell cultures at any scale. Our engineered solutions for sample processing support the analysis of biologics, including cell and gene therapies, to provide enhanced process insights and shorten drug development timelines. As a global leader in innovative fluid technologies, IDEX Health Science provides customers with a comprehensive range of products and tailored fluidic solutions to address process challenges and streamline analytical workflows for bioproduction, while partnering with equipment manufacturers to bridge the gap between cell culture and analytical instrumentation by simplifying and automating sample preparation and delivery. We look forward to partnering with you and leveraging our core technologies, fluidics expertise, and application knowledge to help address resource constraints, accelerate instrument development cycles, and mitigate risks for your next product. In closing, my colleagues and I would like to thank you for attending our webinar today. We hope that these examples have demonstrated how IDEX Health Science fluidic capabilities can help automate and accelerate your bioanalytical workflows. With the IDEX portfolio of products and design expertise, we are ready to partner with you to develop a customized fluidic solution or integrated subsystem to meet your project needs. If you would like to speak with our fluidic experts, explore a custom project, or learn more about our approach to partnership, a process we call IDEXology, we invite you to visit our website at www.idex-hs.com. Thank you.